Good evening, everyone, or good afternoon or good morning, depending on where in the world you are. My name is Chris Bedford-Gay. I'm chairman of FOP Friends in the UK and board member of the International FOP Association. Thank you all for participating today in our webinar. Today's webinar will discuss further the Regeneron LUMINA-1 clinical trial for FOP. This will be looking at the safety and effects of RGN-2477, which is a drug to potentially treat FOP. Giving the talk today is Dr. Richard Keane from the Royal National Orthopaedic Hospital in London. And Dr. Keane is also a trial site investigator for the LUMINA-1 clinical trial in the UK. Presenting today is Dr. Scott Mellis, who's VP of Early Clinical Development and Experimental Sciences at Regeneron. Both will be available for a Q&A portion of this webinar at the end of the presentation. Also joining us is Adam Sherman, who is Research Director at the IFOPA, who will be facilitating our Q&A session at the end. A couple of housekeeping items before we get started. You will be able to submit questions through the Q&A function on the webinar. The button for doing that is found at the bottom of your screen. Questions will be held at the end of the talk and will be asked anonymously. We will be recording this webinar for anyone unable to attend. I think that's the housekeeping and everything else done, so I'd just like to hand over the presentation now to Dr. Mellis from Regeneron. Thank you very much.
Well, hello. Good evening, everyone, and it's a pleasure to be speaking with you today. Can everyone hear me okay? Colleagues on the line? I'll assume the answer is yes. Please let us know if there's a problem with transmission. So today we're going to speak to several topics. Dr. Keane, if you would please advance the slide to the first slide. Before we get to the outline, we'll go through a little disclosures. I am an employee and shareholder of Regeneron Pharmaceuticals. Dr. Keane, would you like to mention yours?
Yeah, I'm a consultant working in London, but I'm a principal investigator both on the Regeneron trial, but also on the Clementia clinical trial as well.
Okay, thank you very much. Next slide, please. So we hope over the next period of time, we will discuss a bit about the clinical challenges of FOP, some exciting laboratory research that's come out of Regeneron Labs that has led us to this clinical development program, a brief thought about clinical research, and then Dr. Keane will be telling us all about the LUMINA-1 study, the anti-activin A clinical trial. Next slide, please. As I'm sure so many of you are all quite familiar, there are some very important challenges associated with fibrodysplasia of ossificans progressiva. And most high on the list for many people are the flare-ups and the heterotopic ossification. So the flare-ups, that can be said to be the fibrodysplasia or the abnormal growth of fibrous tissue in soft tissue. And these are characterized by swelling and pain and stiffness. And very often, those flare-ups seem to be a precursor to heterotopic ossification. That is the formation of new bone where it shouldn't be. That can cause reduced motion, altered growth, and pain. It is also known, however, that heterotopic ossification can occur in people living with FOP even in the absence of an obvious flare-up. So both of these problems are occurring. They can be painful.
Boss.
Now I'm unmuted by the host. Very good. I'm back. Thanks.
Good.
Sorry about that, everyone. Could we have the next slide, please? Dr. King, would you advance?
Yeah, I'm just trying to, don't know what's happened there. There we go, sorry.
Okay. So, recently, and we're talking a few years ago, a mouse model that was genetically engineered to have the human mutation that causes FOP, most commonly, the R206H mutation in the receptor that was identified by many colleagues working with Dr. Kaplan, some of them in the UK. This particular variant was inserted into the DNA of a mouse in a special way so that the mice could reproduce. And this has become a very important and useful model, and it's been shared with colleagues in academia and industry. And so the Regeneron scientists were working with this a few years ago. At the same time, they were also working with cells that had that same FOP mutation in the ACVR1 receptor. And putting those research together really led to important insights, particularly about the role of a protein in the body called activin A, and how that seems to be a very important driver for the manifestation of FOP in people living with the condition. We are going to talk about mice data, and ultimately the most important thing is to test this in people, but we're very enthusiastic and very encouraged by these results. Next slide, please. Okay, so in this mouse model, where now the genome of the mouse looks a bit more like the genome of a person with FOP, that is in the chromosomes, this mouse develops, you're seeing an X-ray of, on the left, the lower portion of a mouse body, and on the right, the upper portion, the base of the neck and the upper spine. And in yellow, colored in with a magic marker, that's false color, it's just colored in to identify where the new bone formation, the new bone that shouldn't be there, labeled HO for heterotopic ossification is. And you can see this looks like the kind of heterotopic ossification that affects people. On the left, it's bridging between the upper leg and the lower leg and the base of the spine, and it's causing immobility, similar to what affects some people with this condition. And on the right, it's a massive bone that is affecting the upper neck and upper spine and base of the skull. So the mouse model seems to faithfully replicate the human disease. Next slide, please. The experiments that had been done in the Petri dish using cells first identified the phenomenon that this protein called activin A would stimulate cells in a way that was very unexpected. And it did not stimulate cells that did not have the FOP mutation in them. This activin A uniquely stimulated bone formation. And so the activin A was the protein from the body was tested in the mouse model. And so when a little sponge that had the activin A protein was implanted into the thigh muscle of a mouse that had the FOP variant, bone formed, and you can see that with the red arrow. When it was implanted into a mouse that did not have the FOP mutation on the right, there was no additional bone formed. So what it says is that the FOP variant, the R206H, leads to a response to activin A that is different from the mouse that does not have that variant. Next slide, please. Okay, so what this led us to hypothesize, pardon me, was that activin A was kind of, we'd call it a bad actor in FOP. And when we deal with proteins that seem to be bad actors in various diseases, we try and think of ways, how can we have the body get rid of that protein? And if you wouldn't mind returning to that previous slide, thank you. One very common way that we make medicines against proteins is using antibodies. And antibodies, you can see two different diagrams of an antibody. These are typically proteins that float around in our bloodstream and help us fight infections. However, in the field of therapeutics, we now make antibodies in a special way that are targeted against proteins that we want to reduce the activity. So we actually had an antibody to activin A already, and this was tested to see that whether the antibody would inactivate the activin and whether that would prevent the FOP processes that were happening in the mouse model. Next slide, please. Okay, so the questions that were being tested were, would the antibody prevent heterotopic ossification from forming in the first place? In the setting where the heterotopic ossification in the mouse was already ongoing and there was already bone there, would it stop the progression? And then thirdly, would it lead to the reversal of some of that heterotopic ossification? Next slide, please. Okay, so basically in what we call the prevention model, basically when the antibody was given to a mouse that had the FOP mutation, and the mouse had not yet developed any heterotopic ossification. In the presence of antibody, those animals, and this was done in a lot of animals, did not develop any abnormal bone. Animals that received an antibody that was not directed against activin A, they developed heterotopic ossification just like their litter mates. If we could have the next slide, please. The other question was, well, if new bone had formed, would the anti-activin A antibody stop the progression, that is, stop the growth or expansion of that new bone? And so in this experiment, the degree of new bone that was present in the animal was measured at several time points, and you can see the week markers, the timing down at the bottom, and the volume of the heterotopic bone is on the y-axis on the left. And so between the weeks 0 and 3, no medicine of any kind was given to these mice. And at week 3, they had both developed a good bit of heterotopic bone. However, at week 3, treatment was started. And the animals in the groups with the black line there, they did not receive the anti-activin A antibody, and the volume of their bone, you could see that line continues to go up. That means essentially more bone is being formed. However, the animals that were in the group that is depicted by the red dotted line, those animals, the volume of bone went down. So what this is indicating is that at the very least, the anti-activin A antibody is stabilizing, stopping the progression of or expansion of the heterotopic bone in these animals. And in fact, it even looks like the quantity of the bone is diminishing somewhat. If we could look at the next slide, please. And in fact, in some instances, not all, it seemed to be the case that some of these new bone lesions were seen to become resorbed or go away, essentially, or diminish in intensity in the setting of continuing treatment with the anti-activin A. So here's a mouse that had a big chunk of heterotopic bone, looks pretty solid, looks kind of solid on the left at day 21. But 21 days later, under the influence of treatment with the antibody, it seems to be becoming reabsorbed by the body. Now, this is probably the case that this is a very new bone lesion. It hasn't had the chance to organize itself and solidify, and it doesn't happen with every lesion, but with some lesions, it's very encouraging that we see what appears to be resorption. So we hope that will translate to people. So if I could have the next slide, please. So in summary, when we look at 3 different settings, when given very early, the antibody prevented the formation of heterotopic ossification. When it was given after heterotopic ossification had begun to form, it stopped the progression, and in some instances, it was associated with reabsorption of the new bone lesions. So these were very encouraging results and motivated us to want to bring this into the clinical setting for testing in people living with FOP. Next slide, please. Yeah, so just very briefly before I turn it over, we want to test now whether these very encouraging results will translate into clinical evidence in people. We believe in safety first. That's the most important thing. We also believe in rigorous experimental design so that the answers that we get from this clinical study, we want to know that they can be believed. This drug either works or it doesn't, and we want to have a very clear answer. Everyone involved, the investigators, the staff, the sponsor, the company, us, we're all committed to good clinical practice and rigorous study oversight by safety committees and by regulatory authorities. We're committed to timely and transparent data reporting. And we know this is a serious commitment for everyone concerned, but we believe it's really important that it be studied. And I'm so glad that Dr. Keene is participating with us. And now I'd like to turn the presentation over to him to describe the LUMINA-1 clinical study.
Okay, thank you very much indeed, Scott. Thank you for that very thorough overview of the animal data to date. And I think what's really exciting is that we are in the arena where we're able to test this drug in humans. And essentially, what the aim of the study is to look at the effect of the drug on making new bone, so the heterotopic ossification. We also are going to look at whether there's any effect on reducing the number or the severity of flare-ups. As we said, this is the first time this drug is being given to patients with FOP, so looking at safety, tolerability is very, very important. And then also looking at the function and the overall well-being of individuals during the study period. Again, these are important things that we will be capturing because we want to know whether by reducing the effects of the bone formation, that in itself will make people feel better, function better. So the study is being conducted in a number of centres throughout the world. It's being looked at in 40 patients. That's the initial target sample size, both men and women over the age of 18. You don't have to have a flare-up to be entered into the study, and if you're having a flare-up, it doesn't exclude you from taking part in the study. And this differentiates it from the Clementia trial, where if you're having a flare-up, you can't be recruited into the study. So, as I say, your flare-up status is not a requirement for coming into the study. You do have to have had some activity related to your condition during the last year. But again, we have found already that most of our patients that we see will report some minor flare or some change in their condition. But again, if your condition has been very, very quiet for a long time, then again, you wouldn't be able to come into the study unless you had a flare, some symptoms. Obviously, being part of a clinical trial involves a number of procedures and assessments. The study involves having a CT scan, sort of with some what's called PET, so some sort of isotope assessment of your bone activity. So you have to be able to have those procedures and be able to sort of accommodate having the scan. But what again we found that the clinical sites are very experienced at looking after individuals with FOP, and we can either on the telephone or with clinical examination basically make sure that you're able to participate and have the scans and a lot of the procedures. The trial also involves having some blood tests, and obviously the medication is given intravenously. Some people might have some concerns about injections, but again, having a blood test or having something into the bloodstream with a drug is not a concern. The injections that we worry about in FOP are obviously the sort of vaccinations, so injections given into the muscle. But for the blood tests and for the infusion of the treatment, that is not a concern. And again, we haven't seen this being a problem with individuals. But again, it's the vaccines into the muscle where there is the concern about those things causing particular flare-ups. So this slide just gives us a scheme of what would happen to an individual when you come in to the study, and I'll just explain it. So on the far left, basically a physician who knows you, or if you go to a site where they will ask you about your condition. So there's basically a sort of screening assessment where people will go over your story with regards to your condition. We will find out if you have the genetic test looking at the ACVR1 gene mutation. You'd have a number of blood tests, clinical assessments, and you can sort of see the sort of the grey vertical arrow showing that during that sort of first 1 month time period, you would have this CT scan with the sodium fluoride PET. This is just a way of looking at the amount of bone that you have in your sort of in the body, and also if there's any activity relating to flare-ups that you might know about or you might not know about. So that initial screening phase takes about 28 days, and then after that you will be randomized to either receiving the drug shown in the sort of green arrow, or you will be on a placebo for the first 6 months. And during that time you will have a monthly intravenous infusion. So it will be that you would come backwards and forwards from the site every month to have your treatment. But then you can see that at 6 months, the top green line stays the same. So you carry on with the treatment for a whole 52 weeks of the year. But the people that have been on placebo will then swap over and then will receive the active drug for the next 6 months. So it's only in that first 6 months where there's people getting a dummy infusion, but then for the rest of the year, everyone gets the active treatment. And again, you can see the grey bars showing when the scans are performed. So there's a scan done after the first 8 weeks of treatment, and there's another scan done at the swap over point when you swap it from one treatment to the next, and then there's a scan at the very end of the So again, so the first half of the study, placebo controlled. Second half, everyone is getting the treatment. And so obviously everyone knows then they are having the active drug. So as I've stated, so the first thing is obviously it does involve coming to the site once a month. You probably would need, at the beginning, as you can see, for that first 28 days, there are a variety of tests. You don't have to be in the hospital for 28 days, but you would just come for some assessments, go home, come back, and then you would receive treatment. But it does involve afterwards probably coming for 1 or 2 days to the hospital site for the infusion and just for checkups afterwards. As part of the clinical assessment, patients have this CAGES score, which is basically a joint score where we measure the degree of movement you have at a number of joints throughout your body. The scoring system is either there's full movement in the joint, partial movement, or the body region is completely fixed. And that scoring system goes from 0 to 30. I've already mentioned the imaging, so the PET-CT is the key critical endpoint of this study. And again, this is a unique sort of assessment that is being undertaken and should give some very, very informative results regarding the efficacy of the treatment. We talked about the medication being given intravenously. The infusion time takes about an hour. But again, as I've stated previously, having an infusion into the vein or having blood tests is not sort of a concern with regards to causing flare-ups. There are blood tests that will need to be done, again, from a safety point of view to make sure that you're well. There's no concerns about other body systems. But this is, again, is a routine requirement for all clinical trials to make sure that individuals are well and healthy. And then subjects, again, will be expected to complete a number of questionnaires. These are predominantly relating to your quality of life, what you can do, what aids you might need to mobilise and help you with everyday daily activities. But again, some very basic questionnaires just regarding how you feel, what you can do. So again, I think with any clinical trial, it's obviously very exciting when you have a new drug to assess in individuals. But again, we obviously don't know whether what's been seen in the animals is going to be exactly the same as gets seen in humans. So we obviously have a duty to monitor individuals, to check for safety, and that's something that we will be looking at as individuals go through the study. I think if anyone listening to the webinar today thinks this would be something they would like to participate in, they will be able to sort of contact a local site. Again, it may be that there isn't a site in your particular country, but there is the option and the possibility that with appropriate discussion between the clinical site and you, it may be that individuals are able to travel to another country to have the treatment, then return home and then go back in. This would have to be taken on an individual basis. But also, before you participate in the trial, you would be sent information regarding the study. You'd be able to read all about the procedures, what would happen, and any concerns or questions you would have would then be dealt with by the clinical team, who would be able to answer all your questions about the study and what would actually happen to you. This would be something that you would talk about on an individual basis with the study team. And again, from after the webinar today, obviously both me and Scott will be very happy to answer any questions you might have regarding either the preclinical data or information about the clinical trial and what would actually happen. Looking at More information about the study is available on the clinicaltrials.gov website, which is available on the screen for you to see here. This both gives information about the entry criteria to the study, some of the background information, but also will list the sites that are actively recruiting at the moment worldwide, and will give you contact details if you would like to sort of contact them and make inquiries about whether or not you would like to participate in the study. So again, I think with any study, you know, as a team of researchers, both clinical and basic scientists, you know, this is a big team effort. There's a lot of, obviously, people that have been involved in the preliminary work, but also without the support of IFOPA, the national sort of FOP advocacy groups such as FOP Friends in the UK, the patients, their carers and their family support members, and all the clinical trial sites who are going on this exciting journey. So again, this is just a list of all the people that have been involved and the people we'd like to thank in getting to the state we are at the moment with regards to moving this clinical trial forward. And obviously, these are very, very exciting times. So I think that's the end of my presentation. I'll hand back to Chris, and then we'll deal with any Q&As that people might have.
Thank you very much, Dr. Keane and Dr. Mellis. Appreciate your time today and giving this presentation to us all on the call. I can see we have a couple of questions that have come in, so I will hand over to Adam to put those to you guys. Thank you very much.
Yes, good afternoon, good morning. So the first question is related to the timing of study results, and roughly when do you anticipate to share the initial results of the Regeneron study?
I mean, from my understanding, obviously this is we have to complete recruitment, so we have to get the 40 subjects into the study, and then we have to have the 1 year follow-up. Obviously, you know, there may be some interim analyses, and I suppose it will probably relate then to just what size effect you might see. So it may be that if the effect is bigger than was anticipated, you might not need 40 subjects. But again, I think at the moment, you know, it will be a little while down the line until the study finishes recruitment.
And is there a expectation for how the study results would be reported back?
I think obviously as soon as any results looking promising, I would imagine that that they would be probably fed back to the individuals who have been participating in the study, because obviously without them, one would not have been able to achieve anything. But then obviously after that, they would then be released, you know, either through sort of scientific presentations or through IFOPA, you know. So I think as soon as there is a positive result that looks good, this would be shared with the wider community. I think then obviously there would be this then would be forming part of the next step with regard to getting, you know, the drug registered, and so that, you know, there may then be further studies, or it would depend on what the regulatory authorities looked at with the outcome measures that were being assessed. But I think as soon as there's some positive results from the clinical side, we would obviously be wanting to move it forward very quickly. And I think obviously from the Regeneron side, they also would have the same wishes.
The next question is, what is the clinical, not scientific, difference of Regeneron's approach with Regen-2477 and Clementia's palovarotene in terms of effect, application, and usage?
So the clinical I mean, the trials are different in the way that with Clementia, the outcome measure you're assessing is different in the fact that Clementia just using whole body CT to look at the amount of bone, whereas in the Regeneron study, you are capturing information about the volume of bone, but because of the sodium fluoride PET, you're getting information about activity and perhaps seeing whether it's changing, making flare-ups become less active or become less frequent. So I think there's a difference in the actual outcome measure that's being assessed. I think also when you look at the sort of the process, it's the Regeneron molecule is working higher up in the cascade, so it's perhaps working higher up, whereas with the so it may have an effect on flare-ups intensity, whereas with the palovarotene, you're looking more down at the end, the flare-ups already happening, and you're stopping this process develop into ossification. So they are different in the pathways as to where they actually work, and the outcome measure that's being tested is slightly different.
Okay, the next question is regarding the HO, and was the HO that was seen in mice models where it was removed or reduced, was that seen under LUMINA-1?
We haven't as yet say that those preliminary data suggesting in the animal data, that hasn't yet been, we haven't reached that stage in the clinical trial to date. I mean, the number of subjects probably actually is yet having sort of their sort of 8-week and their 6-month scans. We haven't got that data to be able to say that yet, but obviously it's promising results. I think as Scott said, it wasn't seen in all the animals who were given the drug, but again, that's something that the trial will be looking at. But as yet, there isn't any data in that effect at the moment.
Just on that same topic, I think the question may have been, have you actually tried removing HO in the mice models, and what was the result? Did it come back? Did it stop? That kind of thing.
Yes, hello, this is Scott Nellis. Yes, we have done some initial experiments in surgery models, and in the presence of the anti-activin A antibody, there is less of a heterotopic ossification response to the trauma associated with surgery as compared to no active antibody treatment. So the surgical data are encouraging. They're still ongoing, and it is our hope that eventually, if we see favorable activity in stopping heterotopic ossification in people living with FOP, that study of the drug in the surgical setting will be forthcoming.
Okay, the next question is two-part. First, in terms of toxicity, was the antibody tested in healthy individuals? And if so, what happened to the healthy individuals when activin A was inhibited?
Scott, do you want to answer that? I mean, I think that's probably better from your side to answer that if you can.
Yes, a phase 1 study was conducted in healthy volunteers, and it was generally well tolerated. The most common side effects were those seen in basically most phase 1 clinical trials: headaches, nausea, diarrhea. You know, none of them interrupting therapy or preventing people from continuing on the medicine. So it was generally well tolerated in this population. That study is what essentially laid the groundwork for additional clinical research in the FOP population.
Okay.
Yeah, most of the adverse events that were seen were mild.
The last question that we have so far is how many clinical trial sites are open and where are they?
So, I mean, I'm just looking here at the clinicaltrials.gov website. There are sites in the US, and I think that's in Mayo and Columbia. But again, the Regeneron team might be able to advise me on the US sites. But I think that in Europe, we have Italy, Netherlands, and the United Kingdom.
That's correct. Italy, Netherlands, the London site, and Mayo Clinic are open at this point. There are additional sites in the U.S. That we hope to be open soon, and other sites around the world as well. So as soon as those sites are open for enrollment, we will let the community know.
So we have one additional question that just came in. If successful, will the trial be opened to children?
I think that, yeah, and I think this is something, again, that as, you know, from both the commercial, from the Regeneron side, but also as us as clinicians, I think this is something that is an active area of sort of work, and that as long as one can be happy with the safety and from some of the preclinical data, I think there will be a move to get the drug to be able to be used in sort of younger children. Because at the moment, yes, the entry criteria are you have to be over 18, but I think it will be probably before. I don't think it'll be the situation where the whole trial has to complete before it gets allowed. It's used in younger individuals. So I think it's very much a sort of a work in progress that will happen at some point, but it will it needs to be done in a timely fashion with the appropriate sort of information going through the regulatory authorities, the IRBs, just to make sure that everything is in place. But yes, I think it's something that will happen in the not too distant future.
So that's the last question that I have. If you do have any, if the people dialing into the webcast have additional questions, please submit them through your Q&A function. We do have some time for additional questions if there are any.
I'm going to dive in here. This is Chris. Just could somebody perhaps speak to the safety of how the drug is administered, so IV. As a community, of course, we're always told that needles are a no-no for FOP families. So maybe you could speak to the safety of that method of delivery.
I'm happy to do that, Chris. Yes. So I think, yes, you're right. I think within the community, you know, everyone is very nervous about injections or blood tests, and I think that that's mainly on the basis of, you know, as I said in my little presentation, that we obviously, you know, having vaccinations intramuscularly or medicines given into the muscle via injection is not a good thing, and, you know, can precipitate a flare. So those are complete no-nos. But obviously we know that you can have vaccinations given subcutaneously, and that can be a sort of that can be okay. But with regards to having blood tests, I think as long as the blood test draw is relatively you know, straightforward, that shouldn't be a particular problem. And again, we've not had any concerns in that regard. So I think as long as the individuals taking the blood tests are trained, competent, then the actual blood test itself doesn't cause a problem. And essentially, you know, having the infusion is essentially just putting, a very small little plastic cannula into the vein. And again, if that is done well, but then there are problems, and we haven't seen that. So I think giving the treatment as an intravenous medication in this trial setting is not a concern. And again, so I don't think individuals should be worried about that aspect of it at all. Does that answer your question?
It does, yes. Thank you very much.
Right.
So, Chris, that is all the questions we have.
Okay. I guess in that case, if there are no more questions pending, I'd just like to extend a thank you again to Dr. Keane and Dr. Mellis for giving the presentation this evening, and to Adam for giving me a hand with my first ever live webinar. I think it's gone reasonably well. I'm sure we will do more in the future, and a thank you to everyone who took time out of their evening or day to participate and ask questions. Unless there's anything else, I think we can probably sign off. A quick reminder that we have recorded this, and we will be sharing this via the IFOPA website and via FOP Friends also. Now, thank you very much.
Thank you all.
Thank you all.